If you like my videos, then don't forget to subscribe my channel and you can share to your friends as well. In today's video, we will discuss about the integral proteins that are present in cell membrane or plasma membrane. We will also discuss about the structure and functions performed by these integral proteins and their type as well. So without further ado, let's get started. Integral proteins. Let's discuss this with the help of the diagram. So you can see over here, these blue colored, this, which are embedded structures are integral proteins. Okay, these are integral proteins. They look like completely immersed in the lipid bilayer. See, these are looks like that they are immersed in this lipid bilayers. Okay, they are having both hydrophobic as well as hydrophilic regions. See, the area that spans the hydrophobic core of lipid bilayer is also hydrophobic regions of a protein, and the area this and this which faces at the cytosolic and or at the extracellular ends is hydrophilic regions or polar region of proteins, because it is made up of polar amino acids. That's why these integral proteins are also known as amphithetic. Okay. The middle part of these proteins that are immersed inside the hydrophobic core of lipid bilayer is composed of, this one is composed of non-polar amino acids. Okay, non-polar amino acids. Therefore, this portion is associated strongly with non-polar fatty acid tails of lipid bilayer. Okay. Besides this, they are also having hydrophilic region, this, on both sides of the lipid bilayer, here and here, that associated with the polar head of lipid bilayer. So we can say that these integral proteins are strongly associated with the lipid bilayer. Okay. So integral proteins are embedded within the lipid bilayer. These bind to the membrane extensively and span the entire width of the lipid bilayer. Therefore, they are known as transmembrane proteins. Why these are known as transmembrane proteins? Because they span the entire width of cell membrane. Next. So let's take another picture to get understand well. So these all four are integral protein types. Some span the entire lipid membrane, okay, at once, and some forming helices and span multiple times like this. We're seeing this particular protein spanning the lipid bilayer multiple times, and this one spanning only at once, and some forming channels like this protein forming channels, and this type of proteins are also known as channel proteins, which are having the hydrophobic tunnel. This is having hydrophobic tunnel. And some are attached to only one lipid layer of cell membrane like this. This particular protein is attached to either inner membrane of cell or either at outer membrane of plasma membrane. So this type of proteins which attached to only one leaflet of cell membrane is known as monotopic integral proteins. So the type of protein that are attached to only one single lipid layer, either with the inner membrane or sometimes at the outer membrane, and they do not span the entire lipid bilayer. Okay, these are known as monotopic integral proteins. The examples of this type of monotopic integral proteins are prostaglandin S2 synthase. Examples is prostaglandin H2 synthase. Basically, it is an enzyme that help in the formation of prostaglandin H2. Next. So I hope you understood why these integral proteins are known as transmembrane proteins, because they span or cross the entire lipid bilayer. Now, on the basis of how many times these proteins span or cross the lipid bilayer, they are classified into two types. The first one is single pass protein, or also known as bitopic proteins, as they span the entire lipid bilayer only at one time. And the second one is multipass proteins or polytopic, as they span the lipid bilayer more than once. You can see over here, these particular proteins are spanning multiple times in the lipid bilayer. Okay. Now next. Now the alpha helical and beta barrel configuration. As we know that alpha helix and beta pleated sheets are secondary structures of protein. The integral proteins that adopts an alpha helical configuration, this is in this form. If it occupies this alpha helical arrangement, then it is known as alpha helical configuration. During spanning the lipid bilayer, basically all types of biological membranes are having alpha helical configurations. The example includes bacteriorhodopsin. It's present in archaebacteria. It uses light to establish proton gradient. It occupies mostly alpha helices that contain non-polar amino acids. Basically, this particular reason, most of the helices is composed of non-polar amino acids that interact strongly with the non-polar or hydrophobic core of lipid bilayer. Next, the integral proteins can have beta barrel configuration where protein occupies beta pleated shapes. It is exclusively present in chloroplast, mitochondria, and outer membrane of gram-negative bacteria. Example of beta barrel proteins is porins. It acts as a channel. Basically, porins are example of channel protein. So the porins acts as a channel. It consists of beta sheets. You can see over here, these are the sheets. Okay. This is the secondary structure of protein. Beta sheets having alternate non-polar and polar amino acids. The non-polar amino acids interact with non-polar tail of phospholipids and polar amino acids forms the interior of the channel. So this beta plated sheets are having non-polar as well as polar protein amino acids, as well as polar amino acids. The polar amino acids and non-polar amino acids are alternatively arranged. The non-polar amino acids interact with the non-polar tail of phospholipids and polar amino acids forms the interior of the channel, means the tunnel of the channel is completely made up of polar amino acids. So it allows the polar molecules to come across the cell membrane without any difficulty. Next. So, transmembrane proteins are the only class of proteins that can perform functions both inside as well as outside, because they span the entire lipid layer, which is present. Some part is present at the extracellular space, okay, and some part is present at the cytosolic space. So that's why these are known as transmembrane proteins, and they act functionally inside the cell as well as outside the cell. The transmembrane proteins have hydrophobic part that are associated with hydrophobic core of lipid bilayer. As a result of these, transmembrane proteins get stuck permanently in cell membrane and are very hard to isolate. So they are amphithetic in nature. They are having both hydrophobic part as well as hydrophilic part. So they get stuck permanently in the cell membrane and are very hard to isolate them. They can remove from the cell membrane with the use of harsh detergents that destroy the lipid bilayer. Once the lipid bilayer is destroyed, then they can only get isolated. That's all for today, and if you have any query, please text me below and share your suggestions about the topics you want me to cover in upcoming videos. Thanks for watching.